Now we will discuss the rest of the exotoxin part 2. In this segment we discuss how these exotoxins of different bacteria work, their mode of action. So there is a very famous exotoxin released from Staph aureus and Strep pyogenes that activate super antigen, which act as super antigen causing shock. So how super antigen cause shock? Super antigen cross links T cell receptor other than the site of antigen binding protein, cross-link TCR with major histocompatibility complex 2, which can cause over stimulation of T cell, which releases interleukin 1, interleukin 2, interferon gamma, TNF alpha, and it can cause shock, which has the features fever, hypotension, rash, permeability, edema, etc. So who produces superantigen? Staph aureus. It produces superantigen via toxic shock syndrome toxin 1. It can also cause food poisoning and scalded skin syndrome, but that will be our next discussion. First we discuss how exotoxin works. So this is the main exotoxin. Then streptococcus pyogenes, it also causes superantigen like feature, but with different toxin, that is erythrogenic exotoxin, and it can cause non-specific hemolysis, not hemolysis, actually it is related to that, but here we discuss the super antigen. So it can cause toxic shock syndrome like features. Then we discuss the next important exotoxin which inhibit neurotransmitter release. So overall there are two types of neurotransmitter. One is inhibitory, which is glycine GABA, and other one is stimulatory like acetylcholine. So how these two neurotransmitter release inhibited by a protein known as metalloprotease, which cleaves snare protein. Snare protein is important in the neuromuscular transmission, neurotransmitter release, and it is important in the last step of when the vesicle neurotransmitter containing vesicle fuse with the cell membrane and exocytose. So there, this metalloprotease, which is also formed, which metalloprotease? There is two metalloprotease, tetanospasmin and botulinum toxin. The tetanospasmin is a metalloproteinase, cleaves near, prevent or no release of glycine and GABA and acetylcholine, and it is secreted from tetanus, Clostridium tetani, causing tetanus, and it is released from Clostridium botulinum, and it can causes botulism of floppy baby. Then next we have inhibit the exotoxin, which causes inhibition of the phagocytic cell neutrophils phagocytic ability, and it happens by a mechanism in which there is inhibition of GI, or in other words we can say disinhibition of adenyl cyclase, which leads to activation of adenyl cyclase, and it can increase CAMP, and this CAMP is causing the inhibition of the phagocytic ability of the neutrophils, and this CAMP and this inhibition of GI is happened by pertussis toxin released from Bordetella pertussis, causing whooping cough. And the last one is exotoxin causing the lysis of cell membrane. So in order to lyse a cell membrane lysis, we need a cell membrane some protein or some enzymes which can act on the cell membrane. So a cell membrane is majorly composed of phospholipids, so we need phospholipase or lecithinase or some proteins which degrade cell membrane. So phospholipase or lecithinase is formed from Clostridium perfringens. It goes to the muscle and cause myonecrosis, and goes to the RBC, can cause hemolysis. And this toxin, Clostridium perfringens, produces a toxin known as alpha toxin. Then we have a protein which is degrading cell membrane, and this protein is formed by Streptococcus pyogenes, and Streptococcus pyogene form a streptolysin. Strepto from the Streptococcus, lysin mean it lyses the cell membrane. So streptolysin, and it can causes it streptolysin as anti-streptolysin O antigen. Streptolysin O is used to diagnose rheumatic fever, and streptolysin is involved in beta hemolysis. So this is overall the discussion of exotoxin. Next we will discuss classification or algorithm of gram-positive bacteria.